You know, we all know the goldfish, right? Maybe your first pet.
That classic fishbowl resident.
Exactly. Well, today we're going to look at it completely differently. Prepare for a bit of a mind shift. Because it turns out this humble fish is giving us some really amazing clues about evolution and how life, well, develops.
It really is. And for you listening, folks who are curious and like to get straight to the interesting stuff, we're going on a journey today that's hopefully really informative, but also, genuinely engaging.
Yeah, we'll skip the super heavy jargon.
Right. We want to make it clear and fascinating.
So our mission here is to explore how studying goldfish embryos, especially when you put them side by side with their cousins, the zebrafish is actually teaching us fundamental things about how life evolves and how organisms are built from scratch.
And here's something, even if you're, say, a professional goldfish breeder or just really into goldfish, we think there might be some fresh angles here on how that human-driven selection has shaped these fish over time.
We hope so. So what's guiding us today?
Our main source is a really detailed research paper. It's called Embryonic Development of Goldfish, Carassius auratus, a model for the study of evolutionary change in developmental mechanisms by artificial selection.
Quite a title.
It is, but it lays it all out. We've pulled out the key insights to talk through.
Okay, let's dive in then. Why goldfish? I mean, out of all the creatures out there, what makes them such a good model for studying evolution?
What's fascinating, it really comes down to their history. We're talking about like 1,000 years of domestication and selective breeding, starting way back in China.
1,000 years. Wow.
Yeah, it's a long time. And over that period, people have bred this incredible range of varieties. Think about different body shapes, weird and wonderful eye types like bubble eyes or telescope eyes.
Oh yeah, the telescope eyes.
Right. And all the colors, different numbers of fins, longer fins. They group them into maybe 16 different types. It's just a huge spectrum, all driven by what breeders like the look of.
It's like evolution on fast forward, but guided by human taste. So how does that link up with, the bigger picture of evolutionary science?
It puts goldfish right alongside other classic examples people study for selection, like dogs or pigeons. You see all this variety within one species, and it makes you ask a really fundamental question. How do the developmental processes, the biological instructions for building the fish, actually change under that kind of intense focused selection?
Right. That's the key, isn't it? It's not just what genes get passed on, but maybe how those genes build the embryo has been tweaked over time because someone wanted, say, fancier fins.
Precisely. That's the Evo-Devo connection, evolutionary developmental biology. And practically speaking, goldfish are pretty good for research, too. You can buy them easily.
Sure, they're everywhere.
And a single female, even a relatively small one, maybe 10 centimeters, can lay hundreds of eggs. Maybe 500 to 1,000.
That's a lot of embryos to study.
Exactly. Gives researchers plenty of material to work with.
Okay, so the variety makes them great for studying selection, but the paper emphasizes comparing them to zebrafish embryos. Why zebrafish?
Yeah, that's super important. Zebrafish are like the superstars of developmental biology research. Scientists know a ton about how they develop, and they have all these amazing molecular tools to study them.
Okay, established model.
Right. And the crucial thing is, goldfish and zebrafish are closely related, phylogenetically speaking. They're evolutionary cousins, sharing a relatively recent common ancestor in the fish family tree.
Got it. So they're similar enough.
Similar enough that a lot of the techniques worked out for zebrafish can be used on goldfish, too. The paper mentions studying things like where the future sperm and egg cells come from the germline, or how the retina develops in the eye. You can adapt the zebrafish methods.
That's efficient. Like having a detailed manual for a slightly different model of car. So what was the main goal of this specific study we're looking at?
Their main aim was to create a really careful, reliable description of how a normal or wild-type goldfish embryo develops. They used a strain called walk-in, which has a single tail fin and looks more like the ancestral goldfish before all the fancy breeds emerged.
Just setting a baseline.
Exactly, a reference point. With that baseline, you can then compare it properly to the well-known zebrafish development, and you can compare it to how all those different fancy goldfish breeds develop. See where the paths diverge.
Makes sense. How detailed did they get?
Very detailed. They defined 7 main periods of development and broke those down into 34 specific stages. And importantly, they link these stages back to the existing staging system for zebrafish, so people can easily cross-reference.
34 stages. Okay. Did they find big differences compared to zebrafish, or was it pretty similar?
Well, this is interesting. Overall, the basic body plan and the sequence of events from the fertilized egg right up to hatching looked remarkably similar, but they definitely spotted differences. Things like the yolk goldfish have a bigger yolk. The way the cells spread over the yolk, which is called epiboly, was a bit different. Pigmentation patterns weren't identical, and the timing, the developmental rate, varied slightly. But the fundamental morphology was conserved.
That's fascinating. So the basic blueprint is stable, but the variations we see in adult goldfish must come from tweaking parts of that process. Let's maybe walk through some key stages they looked at. Where do we start?
Okay, let's start right at the beginning. The zygote, the fertilized egg, and the first cell divisions called cleavage. One thing they noticed straight away is the outer layer of the goldfish egg, the chorion. It's sticky.
Sticky.
Yeah, it makes the eggs clump together and stick to things. A bit different from zebrafish, and it's less transparent, kind of whitish, making it a bit harder to see inside initially.
Okay. And the cell division?
That's called cleavage. Like in zebrafish, it's meroblastic. Only the little cap of cells on top of the yolk divides, not the whole egg. The first division happens pretty fast, maybe 40 minutes after fertilization if it's around 24 degrees Celsius. Then new divisions happen every 25, 30 minutes or so.
A quick start. Then comes the blastula stage, right? That ball of cells.
Right, and here, the bigger yolk in goldfish made things a bit tricky. Defining the standard zebrafish blastula stages, like the 1K cell or dome stage, wasn't as clear-cut.
Because the yolk gets in the way.
Sort of, it changes the geometry.
Yeah.
But they found ways around it, using specific microscopy techniques like dark field and focusing on the edge where the cells meet the yolk. And they saw a neat little difference. At the sphere stage, the goldfish embryo looks kind of like a compressed pear.
A compressed pear, not round like the zebrafish.
Not quite as spherical, yeah. A subtle goldfish signature early on.
Interesting. Okay, moving on to gastrulation. That's a big one, forming the main tissue layers. Any challenges there?
Yes, gastrulation brought up that yolk issue again. Zebrafish are often staged by percent epiboly, how far the cap of cells has spread over the yolk. But the goldfish yolk is softer, bigger, and varies more between eggs.
So that measurement wasn't reliable.
Less reliable, yeah. So they added another measurement,
Yeah.
percentage of blastopore closure. The blastopore is this opening that forms.
Sure.
Looking at how much that had closed relative to the yolk coverage gave them a more consistent way to stage goldfish during this really dynamic period.
Clever workaround,
Oh, yeah.
adapting the method to the organism. What about segmentation when you start seeing the body segments, the somites?
There, things looked much more similar to zebrafish, morphologically speaking.
Yeah.
The somites, those blocks that become muscle and bone, form at roughly the same rate, about 2 per hour.
Okay.
They did notice, sometimes in early segmentation, a little bit of yolk might be exposed at the tail end in goldfish, which, again, probably relates back to the yolk characteristics.
But yolk keeps popping up. Next is the pharyngula period. What happens then?
In zebrafish, the pharyngula stage is often defined using prim stages, based on tracking little clusters of cells called the lateral line primordia, which become sensory organs.
Okay.
But in goldfish, it's harder to see those clearly because the embryos are less transparent, a bit thicker, and the yolk is still a factor. So direct prim staging was tough.
So another adaptation needed.
Exactly. They use something called otic vesicle closure, or OVC. The otic vesicle becomes the inner ear. By carefully observing changes in its size and shape, they could define stages within the goldfish pharyngula period.
Watching the ear develop, basically?
Pretty much. They also noted some differences in pigment patterns like dorsal and ventral stripes appearing differently than in zebrafish, and found that the mouth seemed to open a bit earlier in goldfish development.
It's amazing how these subtle early differences might connect to the features readers selected for later on. What about hatching? Is that straightforward?
Well, hatching time itself isn't a great marker because it varies a lot between individual fish and depends on the environment. So instead of just time, they define hatching stages based on morphology, things like how developed the pectoral fins are, long pecfine stages, pigmentation details, and the angle between the head and the body, protruding mouth stage. And again, they related these back to similar features in zebrafish hatching stages.
Right, focusing on the physical changes, not just the clock. So pulling all this detailed comparison together, what's the big picture implication for evolutionary biology? What did the researchers conclude?
The main takeaway, and it's a big one, is that this work really confirms goldfish embryos are a fantastic, maybe even unique model system for digging into how domestication, that artificial selection process, actually impacts development itself.
Okay.
And because that basic early development is so similar to zebrafish, It means we can potentially use all those powerful molecular tools developed for zebrafish.
And apply them to goldfish.
Exactly. To figure out the genetic changes that underlie all that amazing variety we see in goldfish, breeds the different shapes, colors, fins. It lets us connect the genes selected by breeders to the resulting developmental changes. It's a bridge between genetics, development, and human selection.
That's powerful. You've got this creature sculpted by humans for centuries, and now we can potentially use cutting-edge tools to see how that sculpting happened at the deepest level.
Precisely. And the researchers point towards future work directly comparing the development of this wild-type Viking goldfish with all the different mutant breeds. That could really pinpoint how selection altered specific developmental steps. They even mentioned that some older studies hinted at differences between wild-type and mutant embryos before hatching.
So the changes start really early on.
Seems like it. It gets right down to how evolution, even human-driven evolution, tinkers with the basic instructions for building an organism. They also suggest comparing goldfish development to similar-looking spontaneous mutants in other fish, like Medaka, to see if the same appearances come from tweaking the same developmental pathways. And of course, comparing goldfish shaped by artificial selection to fish with really divergent shapes due to natural selection, like sticklebacks or cavefish. That contrast could be really revealing.
So the staging table isn't just a description, it's really a foundation for a whole lot more research.
That's exactly the hope, that this detailed map will really help researchers do more molecular studies on goldfish and get much deeper insights into how selection, development, and evolution all interact. And for the breeders and enthusiasts, it gives a whole new layer of understanding to the incredible fish they work with, seeing the deep developmental story behind those forms.
So wrapping this up for you, our listener. The common goldfish, maybe swimming in a bowl in your memory or even your living room, is actually this incredibly valuable scientific window. It helps us understand how selection, in this case, human preference, can drive evolutionary change right down at the level of how an embryo is built.
Yeah, that's the real aha moment, I think. Just how much deep science can be hidden in something so familiar. It's not just a pet, it's a living lesson in Evo-Devo.